Good morning. I want to do just a real quick video to go over how to do Punnett squares, just in case any of you are struggling with how to arrange them and fill them in and analyze them. Let's start with a scenario where we're given two genes. Remember that a gene is made up of two parts, two alleles. One that you got from your mother, one that you got from your father. And those two, the combination of those two, actually will determine what your gene is. And we note or we write the alleles or the factors that make up a gene as letters. If you have a capital letter, it's a dominant allele, and if you have a small letter, it's a recessive allele. So we're always going to have a situation where we have two letters, okay, because that's what's going to make a gene, and that gene then is going to make a trait. So let's say we're given a situation where we're talking about pea pod colors, all right? And the dominant allele for a pea pod color is a capital G, and that means that the pod would be green. And the recessive allele would be a little g, and that would mean that, or it would code for a yellow coloring on the pea plant. So let's say we take one pea plant, so we're just going to call it Dad, just because I think it's easier to kind of organize your thoughts that way. And then we have another pea plant here, Mom, okay? So Dad is homozygous dominant, homozygous meaning that the two letters are either both capital or both small, lowercase. So he's homozygous dominant, meaning that he's got two capital letter G's, and we're going to put Dad along the top here. So we're going to basically spread out his alleles, okay? And then we have Mom here. Mom is a homozygous recessive. Again, homozygous meaning that they're the same, and she's two little g's. She's actually a yellow plant. So we're going to put Mom along the side. All right. Now, remember, there's one other possibility that it could be, which would be one capital and one lowercase, and we would call that heterozygous. And in a normal Mendelian trait, this plant would still be green because it's got that dominant allele in it, as long as there's a capital letter in a traditional type of gene, then the dominant trait, which is green, would be shown. So now that you've put Dad along the top and you've put Mom along the side here, you're going to fill in the box. So I'm going to go down capital G, I'm going to go down capital G. You're just bringing them down. I'm going to go down capital G, I'm going to go down capital G. Okay, now go across. Little g, fill it over, little g, little g. Okay, so these are your offspring. These are all of the possibilities that these two parents can create in terms of what their genes will look like. They got one allele from their father, they got one allele from their mother. Okay, Mom and Dad each had two alleles for a gene. All of the offspring have two alleles for a gene. Now, if you look here, we can figure out the probability then. So 4 out of 4 offspring are going to be heterozygous, big G, little g. And that's the genotype, okay, which just means the combination of letters. We also know then that 4 out of 4 are going to be green. This is the phenotype. This is what you actually see. Okay? There is no chance in a traditional Mendelian trait that these two parents are going to produce a yellow child or a yellow pea plant. None, zero. There is a 100% chance, 4 to 4, that their offspring will be green and their offspring will have the genotype that's heterozygous, big G, little g. So let's try another one real quick. Same idea. Big G is green, it's the dominant. Little g is yellow. Okay, let's say we're going to take a big G, little g parent times a little g parent. All right, set up your box. You've got 4 squares in here. Put one along the side. It doesn't matter which one goes on the side and which one goes on top. It makes no difference. Just like it makes no difference what order you put the letters in here. I typically put the capital letter first, so it's easier for me to organize, look at quickly, but it makes no difference. Put this one along the top, little g, fill in. So big G all the way over, little g all the way over, little g down, little g down. Okay, look and see what you got. You have 2 out of 4, or 1/2, is going to be heterozygous. That's the genotype. There's 1 capital and there's 1 lowercase. All right, 2 out of 4, or 1/2, is going to have the homozygous recessive, homozygous because they're the same, recessive because they're the little g, genotype, and they will have the yellow phenotype. So this one would have a green phenotype, because there is 1 capital letter here. There's no capital letter here, so the only way that you would get this trait would be to be little g to get that yellow phenotype. Obviously reduce these, but you know, hey, you can check your work pretty easily by just, hey, 2 plus 2 should equal 4, which is the bottom number. You got them all covered. Piece of cake. So things that get people confused, making sure that you're putting 1 parent along one side and 1 parent along the top. You want to separate out the alleles, so each of these offspring got 1 from 1 parent, here it is, and they got 1 from another parent, here it is. So go through the practice problems, and if you have questions, don't be afraid to ask. Have a great day.